Let's now take a quick look at the concept of a test cross. Phenotypes can be seen, genotypes cannot. You can go to the hutch, grab a straight-eared rabbit. You'll know that that rabbit doesn't have floppy ears, it has straight ears. But the genotype within the nucleus of that organism is not as quickly apparent. So that's where Mendel developed what's called a test cross. The test cross crosses the dominant phenotype with the recessive phenotype. So what we're going to do here, we're going to use purple as the dominant trait, white flowers as the recessive trait. And I want to go a little closer here because the letter P doesn't reveal itself extremely well for what we're looking at. So what we're saying here is that this dominant phenotype could be either homozygous dominant or it could be heterozygous. And by looking at it, we can't tell which it is. But we know immediately that the recessive phenotype has to be homozygous recessive. It can be no other way. Therefore, what we end up with, if we were to cross a homozygous dominant purple flower with a homozygous recessive white flower, I'm going to highlight in red here the recessive alleles. Okay, let's do that a little better. I'm going to highlight in red here the recessive alleles. Okay, so as those recessive alleles in the Punnett square get passed on, the dominant alleles do their thing, and all that can happen is heterozygous genotypes. The only offspring that can be produced then are purple flowers, 100% purple. However, the test cross can reveal what's inside that purple plant. So let's say we have now a heterozygous purple plant, and let's cross that. So now we have, highlighting in red again, here we have our recessive alleles and the recessive allele that was being carried now by that heterozygous purple plant. So the top part of the Punnett square would produce heterozygous purple plants, but the bottom part of this Punnett square now will produce homozygous recessive white plants. So the test cross reveals what's inside that purple plant. If that purple plant is homozygous, we end up with 100% purple plants. There's nothing else that could be produced. Okay? If that purple plant is heterozygous, we get a 1-to-1 ratio. A full 50% of those plants will show the recessive phenotype and reveal then what was inside, what was being carried by that purple plant. The test cross.